And thank you so much for joining us. And I know it's quite late in Zurich right now.
Yes, also very happy to be here, and thank you for the invitation. Can you see my slide, and can you hear me well?
Yep, you're
okay.
coming through there.
So today I will be happy to share with you the most recent developments we have made at ETH at intersection between NMR spectroscopy and machine learning. So nuclear magnetic resonance spectroscopy plays an important role in protein structure studies. So far, it contributed with more than 11,000 structural models to PDB. Moreover, NMR spectroscopy spectroscopy measurements are characterized by very rich information content, which allows to explain protein dynamics and interactions mechanistically at the atomic level. Unfortunately, NMR spectroscopy suffers from very tedious data analysis workflow. Nowadays, it takes weeks or months of manual work of experienced spectroscopists to build a structural model out of set of NMR spectra. This process involved peak picking, where the 2D, 3D, or 4-dimensional volume of the spectrum is inspected visually to identify signal coordinates, which are then mapped to atoms that we know from the protein sequence in the process called chemical shift assignment. And this mapping of signals to atoms allows to extract from experimental data interatomic distance restraints. And based on set of such distance restraints, we can reconstruct the protein structure. Automation of this tedious process is an open problem, long-lasting, that has been formulated already in 1990s. And only recently at ETH, we have proposed the first comprehensive solution. Our method, Artina, takes as input only set of NMR spectra and output peak positions, chemical shift assignment, and structure, strictly without any human intervention and within a few hours of computational time. So it means that considering that we also provide software implementation of the method, the workflow, which was previously very tedious, costly, and time-consuming, can now be executed even by non-experienced person, and in much shorter time, of course. So thinking in terms of prediction task, working on this problem involves with dealing with multiple data modalities. If we look at the left side of the slide, the input data, we typically have set, which consists of variable number of 2D, 3D, or 4D experiments. Typically, this is 10 to 15 NMR spectra, and they are all represented as the regular n-dimensional grid of data. And also, we, of course, use protein sequence represented as just sequence of letters in one letter encoding. Then in the output, we have a graph representing protein, which is folded in 3D space. And for each atom, we predict 3 coordinates of an atom, as well as the quantity called chemical shift, which is strictly linked to NMR spectroscopy and which explains the local chemical environment of an atom. So this visual abstract presents interplay between main components of our Artina method. So the input data, composed of set of spectra and the sequence presented in the upper part of the slide, undergoes visual spectrum analysis, which includes peak picking and the convolution and unfolding. As a result, we have a set of signal coordinates together with some quantification of uncertainty, whether given signal corresponds to true peak coming from our sample or some artifacts which can be caused by protein degradation, interaction with water, or some other issues. Once we have set of signals, we use a FLIA algorithm to map them 2 atoms in the protein sequence, and one which is solution to chemical sheet assignment problem. And then in the next step, we bring to our workflow information about thousands of proteins solved in the past by NMR spectroscopy. It has form of graph neural network, and it allows us to reassess the initially observed spectrum, what closes so-called chemical shift refinement cycle. In structure refinement cycle, we calculate 10 intermediate structure proposals, given the current state of chemical sheet assignments, and we use gradient boosted trees to rank them and pick the best structure as contextual information, which again allows us to reassess the spectrum that we observed. So in Artina workflow, we execute chemical sheet refinement and structure refinement cycle iteratively 3 times until we reach the final output, which is our protein structure with assignments. So to build our method, we had to collect a quite large training data set. Unfortunately, it was a major challenge because such data is typically not deposited in PDB, BMRB, or any other public repositories. Therefore, we decided to establish 4 data acquisition channels. At first, we implemented the website presented in the slide, and we encourage research groups worldwide to upload their published data. Then we implement database crawler, which systematically scanned FTP server of BMRB database and extracted data files relevant to our project. We also explored our collaboration network here at ETH and also internationally, and contributed with our internal resources. So in total, we managed to collect 1,329 2D, 3D, and 4-dimensional NMR spectra, which allow 100 proteins to be reproduced out of the original measurements. So this data set by NMR standards is quite substantial because it corresponds to years of measurement time with very expensive instruments. So this figure presents the summary of the data set that we collected. In the middle, there are PDB codes of all proteins. The upper histogram presents number of spectra we collected for each protein with color-coded groups, color-coded experimental types. And in the lower part, there is information about what type of NMR spectra we collected for each protein, because this project was about structure determination in principle. We have carbon and nitrogen resolved NOSI for every protein, as well as some arbitrary subset of other experiment types. So in order to use this data for deep learning modeling, we had to transform it into a training data set. And to do so, we extracted 600,000 2D patches from collected spectra. And each patch presented in the left part of the slide is centered at the signal in NMR spectrum. And it was also quite tedious work to provide manual annotations for 600,000 patches. And each of them has been annotated, whether it contains true peak or artifact in the central part, which is highlighted in blue. So it was prepared for binary classification task. In addition, we implemented a simple NMR spectra fragment generator presented in the red, which can generate not the entire spectrum, but rather a smaller volume with sufficient approximation of the real measurement process. And the advantage, of course, are that this source of data is almost unlimited. We can modulate difficulty of the fragments to analyze, and annotations are unambiguous, which is not the case of experimental data with manual annotation. So to handle visual spectrum analysis, we trained two compact deep residual neural networks. The first one, presented in the upper part of the slide, takes as input quite large two-dimensional fragment of the spectrum, and its goal is to solve the binary classification task, answering the question whether the signal presented in the central part of the image patch contains a true peak or an artifact. The lower architecture is designed for peak signal deconvolution, and it takes as input smaller but three-dimensional part of the spectrum centered at the signal, which was classified by the first network as a true peak. And the convolution network outputs a matrix 3 by 3, which corresponds to 3D coordinates of up to 3 deconvolved signal components. Because we had to assure some properties of the model, such as permutational invariance of the output, the lower model has been trained with Hampfer loss. So this video presents serial sections through three-dimensional NMR spectrum and the example output of our models, just to explain qualitatively what it involves to analyze their spectrum. In principle, we observe typically quite a few artifacts, which comes from imperfection of the sample preparation measurement process, but also, for example, from interactions of the sample with water. So once we have our signals identified, we still need to assign them to atoms in the protein sequence. And what is challenging about it is that it involves combinatorial optimization and searching the combinatorial space of size for the medium-sized protein, about 1000 factorial. And this problem has been proven to be NP-hard. So we solve it iteratively. So the main method used in this problem is FLIA algorithm. So it works such that it formulates two sets of peaks, expected and observed one. Expected peaks are the ones that we expect to observe in the spectrum, given the knowledge which spectrum we record and which pulse sequence has been used. And in that case, we know the assignment of every signal, but we know its position only approximately because we have some statistical knowledge where we observe certain atoms in the spectrum, but it is never exact until we actually observe it in the spectrum. And on the right-hand side, we have observed peaks, which have known position because we just found them in the spectrum, but we don't know from which atoms they come from, so they have unknown assignments. And the chemical sheet assignment is about mapping expected peaks to observed one. And it is in FLIA algorithm, it is achieved through optimization of the objective function, which accounts for completeness, because we want to assign, in principle, all atoms in two peaks that we observe in the spectrum, alignment of peaks assigned to the same atom, normality of assigned peaks, because we want assignment to be in agreement with some statistical knowledge that we have from BMRB database, and also low degeneracy, because we would like to avoid situation where our method assigns several expected peaks to one observed one. So in order to facilitate the optimization process, the combinatorial optimization is driven by message passing graph neural network. And the idea is as follows. If you look at the left-hand side of the slide, there is a protein fragment. And let's assume for a moment that atoms marked in red, for some reasons, are difficult to assign. Then the procedure that we execute is that first we run the combinatorial optimization in the step 1, which typically gives a very confident and reliable assignment for quite many atoms in the proximity of this aromatic ring. And since there are many atoms which are in quite similar chemical environment, one can postulate that if we know chemical shifts of atoms in the proximity of this aromatic ring, it also gives some information about possible distribution of atoms in the aromatic ring that we want to infer. And here the graph neural networks comes into play, and given in the step 2, given assignment found by FLIA marked by green, we would like to predict distribution of each atom marked by red. It gives some additional constraints and allows for reinterpretation of the spectra and the signals that we observe, and it allows to identify better assignment in the following FLIA assignment in the step number 3. So the models that we use in this particular problem are message passing graph neural networks. So here every atom is represented as a node and every chemical bond as an edge. We also enhance this model by adding some additional edges that corresponds to possible full space contacts. And the general formulation is that we need to define 3 differentiable functions. One is responsible, MVU, is responsible for message generation from node U to V, given hidden state of 2 nodes and edge between them. The second function is called message aggregation, and it combines all messages that node receives from its neighbors into a single vector. And then the final function updates the hidden state of a node given the aggregated messages and the current state of the node. So in Artina workflow, machine learning comes in several places and different forms. I think that covering all of them is beyond the scope of this talk. However, in case such information would be useful or interesting, it is described in the paper that we published almost 2 years ago. So in order to evaluate our method, we used all 1,329 spectra to automatically reproduce 100 protein structures. And then we compared the result of automated chemical sheet assignment with BMRB database depositions and the output of automated structure calculation with PDB depositions. So this particular slide presents evaluation of the chemical sheet assignment. The inner circle presents PDB codes of proteins and which are sorted by size clockwise, and each bar plot presents the accuracy of the chemical sheet assignment. So in total, the median accuracy of the chemical sheet assignment of backbone atoms with our method is 97.2%. What one can also observe that this accuracy is not strictly correlated with the size of the system, which plays a major role in NMR spectroscopy, because typically we study proteins up to 20 kilodaltons, and above this level it gets more tedious. On the other hand, in some sense, this range of system size is complementary to some other methods. So in case of sidechain assignment, this accuracy is around 88%, a little bit lower because of challenges associated with aromatic ring assignment and assigning long positively charged sidechains. overall, our method yields 91.4% of the chemical shift assignment accuracy. So this video presents 100 protein structures solved by Artina, color-coded in blue, which are overlaid with 100 PDB structures, here color-coded in orange. Each of the structures is displayed for 1 second, and they are sorted from the best to the worst prediction. Then in the upper part of the animation, you can find PDB code of the system, RMSD of the predicted structure to PDB deposition, as well as chemical sheet assignment accuracy for backbone and for the side chain. The part of the structure which is highlighted corresponds to the fragment which is well folded, whereas flexible tails or loop are displayed with some degree of transparency, and they were identified with psi-range algorithm. I hope that you will find it a bit convincing that with Artina we can make quite reasonable predictions of the systems under study, and maybe I will just wait a few seconds so that you can look at maybe the most challenging and difficult predictions. So in the next slide, I present the same information, but in more quantitative way. So again, in the inner circle, we have PDB codes sorted by size. And in the left panel, each bar corresponds to RMSD, the predicted protein structure to PDB deposition. Overall, the median accuracy is 1.44 angstrom. And again, we don't see much correlation between the system size and the accuracy. What is also interesting to observe is that out of 100 structures that we use in the study, only 3 of them have RMSD above 3 angstrom. The right panel presents a number of distance restraints. Our method extracts automatically from experimental data per residue. And it is also important information because the more restraints we have, especially long-range ones, the more accurately we can identify positioning of atoms in the structure. So the median value is around 20. This is the number of restraints our method can extract in a fully automated manner. So if we look at the worst predictions, so only these 3 structures out of 100, where the RMSD of the predicted structure was above 3 angstrom, we can observe that in all 3 cases, the problem was associated with positioning the first or the last secondary structure element. So in the Artina structures presented in blue, the alpha helix was at different angle compared to the PDB deposition, whereas the rest of the protein was in agreement, so had very low contribution to this high RMSD value. And to further challenge our methods, we acquired some spectra with only 20% of carbon-13 labeling. So it yields to some experimental data, which is featured by lower signal-to-noise ratio. We observed quite strong background noise and also in artifacts. But also in this case, with this challenging data, our system correctly solved the structure of this protein. If I remember correctly, the overall RMSD was around 2 angstroms. Finally, in order to prove that every component of this complicated Artina workflow is necessary and contributes to the final solution in a positive way, we carried out some ablation studies where we measured all the quantities reported in our work at different stages of the workflow. So once we managed to solve the problem or propose solution of automated structure determination, at least for some systems, we're thinking about a way of making this technology available to the entire NMR community. And we decided to build a platform, NMR Artist, where people can register accounts and upload the experimental data. It works to some extent as simplified Google Drive or Dropbox. So one can just upload some spectra and then run analysis with our deep learning models online without necessity to install any packages locally. So our platform got quite substantial attention from the NMR community. So at the moment, we have approximately 1,000 user accounts registered, and over 2.5 years since the release of the platform, we received almost 20,000 2D, 3D, and 4D NMR spectra, which is also quite substantial amount because it takes between few hours and few days to measure single spectrum. Then one can do some math and estimate how much measurement time was required to upload this data. Our users also carried out more than 7,000 automated chemical sheet assignment. This is maybe also important because chemical sheet assignment in NMR is an entry point for studying dynamics and interactions. And also we have 3,000 structural calculations, which is nowadays maybe less relevant because of other methods, including in silico. However, still we have some cases where studying protein structures with NMR is still relevant. So this map presents public research institutions which have at least one NMR Artist account registered. And we have majority of our users in Europe, United States, and Asia Pacific region. And we basically handle this automated data analysis on the daily basis. So our technology reached level of maturity where we're able to establish partnership with Bruker, one of the leading manufacturer of NMR instruments, to further grow this software and technology, make it easier to use, and also expand it to handle more types of studies that are done with NMR spectroscopy. So, although automating data analysis with NMR is valid and interesting research problem, we ask ourselves, can we go one step further? Can we leverage deep learning to propose a way of studying proteins with NMR spectroscopy, which is not possible to carry out manually, just by manual data analysis? And recently we started a new project which has precisely this objective. And our idea is that instead of recording a series 10 to 15 3D or 4D experiments, we will record just a single 2D experiment with 3 channels, such that information from this vast data set used nowadays will be just compressed to single 2D image. And after this compression is made, the experimental results are no longer human interpretable or barely human interpretable, but we hope that they can be analyzed by deep learning model. And if it works, it has some serious consequences because we can reduce the measurement time with NMR spectroscopy from approximately 2 weeks per protein to just single day. But also we can expand a range of protein systems that you can study with NMR, because the new way of measuring and analyzing data does not assume that we need to make protein expression in bacterial cells. So we can also use proteins just found in some natural sources. Also, by utilizing a new method, we no longer need to use isotope labeling, which is currently costly and tedious in regular workflows. And this method also doesn't work as a black box, because although the data analysis is AI-driven and cannot be carried out manually by human annotator, however, the problem is asymmetric. So although manually we cannot solve the problem, given the solution that we have from machine learning model, we can manually verify against experimental data whether the solution is correct. And we think that this type of project represents deep learning-driven innovation, because the measurement technique has been established so far in the past. However, the problem was more on the data analysis side. So this slide presents some simulated three-channel spectrum that I talk about, and as you can see, we have quite substantial signal overlap, and especially if you look at the region marked in panel B, I think it will be quite convincing that manual analysis of this type of spectra would be rather difficult. And the protein system used to simulate the spectrum also wasn't the largest one. I think it was around 10 kilodaltons. So we plan to approach this problem in end-to-end fashion using vision transformers. So the idea is that our deep learning model will take the entire spectrum as an input, in contrast to smaller models that we trained so far. And it can learn all the rules that we use to analyze NMR spectrum implicitly. And the idea is that for every signal in such input spectrum, the model will predict some annotation linked to atom type, amino acid type, and these partial annotations will be sufficient to reconstruct assignment and the structure using more classical methods. So, so far we have some early success with this new approach. So these two panels present ground truth and predicted protein structures using this three-channel approach. So far, these are quite small systems, even for NMR standards. However, the project grows, and we make progress every month to make it working also for larger systems. So now I would like to make a link to protein dynamics, which is maybe of the core interest in this session. So the first message that I would like to send, it was already mentioned in the introduction, is that NMR spectroscopy offers a wide array of tools that can be used to study system dynamics at many different time scales, ranging from picoseconds to hours. And for every time scale and type of dynamics that we want to study, there are some dedicated experiments and protocols, such that if we think about sidechain rotation and some pico to nanoseconds, there is R1, R2, and hetero-NOE experiments. And the second message that I would like to send is that these experiments linked to system dynamics are the same type of experiments that we use to find assignments and find protein structure. So in principle, these are experiment types that are already supported by our deep learning models. And the dynamic information is encoded in features of signals, such as, for instance, peak intensity. So it means that we are placed in very good position to expand our Artina framework such that on the top of assignment or optionally protein structure, it will be possible to infer system dynamics at all time scales supported by NMR spectroscopy, depending on what data set is provided by the user of our platform. And what is also, I think, quite interesting is that NMR spectroscopy can also explain dynamics mechanistically. So there is this ENUE technique, which we have some expertise in our group. It is called exact nuclear Overhauser effect, and it allows to extract very precise interatomic distance restraints from experimental data. And advantage of such procedure is that if we have so precise restraints, they start to form some clusters that are mutually exclusive. And this way we can infer that certain systems may have two or more states, and only in such circumstance, all these sets of restraints started by ENUE can be fulfilled simultaneously. And example of such study is cyclophilin A. It is work from our group from 2015, where ENUE were used to find two-state model of the protein with open and closed state as indicated in the right-hand side panel. So now I would like to move to some closing remarks. So we established a large-scale benchmark for our open research data initiative, where we collected 1.3 thousand spectra, allowing to reproduce 100 proteins from the original measurements. We proposed the first comprehensive solution to the long-lasting open problem of defining assignment and solving protein structures by NMR. We also established cloud computing platform to our technology to a broader range of users. And now we are working on end-to-end approach with vision transformers that is supposed to go beyond automation. And finally, we are also in good position to think about dynamics and incorporating relaxation data in our modeling. If you are interested in our work, there are 3 publications that I wrote recently. The first one covers machine learning part of Artina method. We also propose a way of integrating our approach with AlphaFold and some NMR in silico predictions like UCB shift. This is covered in second paper, and the third one is about all the benchmark data that we collected in the past. And now I would like to acknowledge my supervisors, Professor Roland Rick and Peter Gintert. I worked more or less independently on the work presented in this presentation as my postdoctoral project. And we received a lot of support from European Commission within Marie Curie actions, Eurostar, ETH Zurich. And recently I was also awarded by a 4-year grant by Swiss National Science Foundation. And it is to grow this idea of going beyond automation with deep learning and NMR spectroscopy. And it is all what I presented for today. Thank you for your attention.
Wow, that was fantastic. Okay, we already have a question.
Hey, Peter, this is Hannah Wayman-Steele. Really fantastic work. I'm a postdoc with Dorothy Kern, and please consider me a very excited future user of all your techniques for exactly what you're saying. So my future lab will do relaxation experiments, and we'd love to be able to, not spend our lives assigning, but rather move on and get to these other types of data. But I haven't been able to use the NMRDIST platform currently because the systems of data that I work with all contain peak duplication for one reason or another. When when there's sufficiently slow interconversion between different states, and this results in multiple peaks. So my question is YA sort of intrinsically set up to handle that? Like, is that a forthcoming feature? Because it's something that I want to use it, but unfortunately, just it's not working for me.
So unfortunately, at the moment, we support only just single state proteins. So if there are two sets of signals, then it causes some problems in the Artina workflow, and one can handle them in by introducing more manual input, because the main effort after the main release of the Artina was to make it more open and more interactive with people. So in case, for example, that there is multi-state protein and it is not handled inherently by Artina, what one can do is one can upload all the data, execute as if it was single-state protein. Of course, the final model will be suboptimal, but anyway, there will be output of every intermediate step, peak positions, chemical shift assignment, and structure. And what one can do is manually correct peak positions by removing, for instance, the second set of peaks that corresponds to second state, and use this manual
a
defined
priori
peak
one
list.
doesn't necessarily know which corresponds to which state, or if there's your cis-trans protein isomerization that's actually another state that you don't care about. Like, to me, that's sort of the bottleneck at the moment for these dynamic proteins. I'm just curious because I don't know, it's a hard problem, but to me, that's sort of the next frontier of how to automatically handle that.
So at the moment, as I mentioned, it is not like directly handled by our system.
Yeah.
But for me, it is always interesting to look at different projects, and I do it quite regularly. So if you are interested, I can also look at your data, and maybe I will learn something how to improve our system further.
Yeah, if you have the bandwidth, I'd love to share an example. Yeah, of this. The system I worked with is this Chi-B fold switcher, and there's 4 macro states, and so it took a number of postdocs quite a while, even though it's just a 17 kilodalton protein. It took a long time to assign.
Yeah, so for me, it would be enough to know the hash code of the project or the structure calculation run, and I would be curious to see how it works in such situations.
I'll send you an email then. Yeah, that'd be awesome.
I guess with duplicated peaks, you get a 2D NMR that's just like an incoherent average of both your individual 2D NMRs. So
it's basically, so if there's 2 that are sufficiently slowly interconverting, there will be 2 sets of peaks in the spectra, and so then it's essentially 2 different molecules.
And you can't experimentally freeze the protein in one of the states.
Well, one can, and that's sort of sometimes how one might, go about figuring out which state corresponds to which. But I would like to be.
It sounds like that you're just seeing a mixture distribution, and you have this problem of figuring out which, doing an inference with a mixture.
Yeah, and so in principle, I do think it's like tractable to inference, but it's just then a matter of what priors are you picking from to figure it out.
I have another question about the dynamics. So the dynamics that you see experimentally, how do they compare to the dynamics predicted by molecular dynamics? Basically, they overlap, or there is a portion that overlap and there is a big fraction that do not, or?
So I think it will be relatively hard to answer this question. It is because my background is in computer science. So I hold PhD in machine learning, and I joined the group in 2019 to develop software and build machine learning models. So I think someone with more expertise in practical expertise in mass spectroscopy would be better to answer this type of questions.
Okay. Thank you.
Can I ask what's the input for chemical shift prediction? Is it just the topology of the molecule?
So for the chemical shift prediction, we need NMR spectra and protein sequence. So so recently we also made an update which allows to define arbitrary topology. It is more applicable to protein ligand complexes, but in principle one can also use now Artina for RNA or DNA, but it is still not very well tested. It is just a possibility. So in case of proteins or RNA, DNA, it is just a sequence. In case of small molecules, then we need to know the structure.
More questions? I have more questions I can ask.
Is there any quantum mechanics that goes into the shift assignment? Because you could, in principle, predict the NMR shift with a QM calculation. It's just really expensive.
Yes, I'm not sure to what extent these predictions are accurate. So in terms of so there is another subfield at the intersection of machine learning and NMR spectroscopy, which deals with chemical shift prediction based on protein structure and about predicting properties of the molecules given chemical shifts. So recently, I even started to write a literature review on this topic. And currently, the dominant techniques are either machine learning approaches, which try to predict chemical shift assignment directly, or some machine learning methods which try to approximate quantum mechanics to make the prediction faster. And I'm not sure to what extent trying to predict them through the QM simulation would be practical in this case.
You thought about using a diffusion model for the shift refinement task?
Diffusion model for the chemical shift refinement task. So in how would you define the usage of diffusion model for the chemical shift refinement in such context?
As I understand it, you have a graph neural network edge there that takes as input the chemical topology and estimates of the chemical shifts and refines them, right?
Yeah.
You could imagine having that same graph neural network as the transition kernel in a diffusion model.
Yes, I think that it will be definitely to replace just more traditional GNN with diffusion model. Obviously, the advantage would be that this prediction will be made in multiple steps, somehow gradually, and there is hope that it will be more accurate, I think.
Well, I was thinking it might be a way to address the peak splitting idea, because then you could have an ensemble of shift predictions rather than a single value for each nucleus.
But in case of the multi-state proteins, so it also depends what corresponds to each state. But in some cases, I think it happens that the peak can move quite substantially from between two states, and therefore to infer which atoms are assigned to each peak. It involves utilization of some non-local information. So I think it is possible, but in principle, such predicted distribution would need to be quite multimodal.
Yeah,